My name is Kate.
And I'm Carly.
And this is the podcast called
You Cannot Be Serious. It's a really horrible pun, but that gives you a little insight as to who we are.
It's even better when you explain it. So, what is this podcast? It is a podcast that we made to talk about cell biology. So, who we are graduate students here at University of North Carolina, Charlotte. So, what is a graduate student? We are graduate students in biology, so that means that we have already gotten our undergraduate degrees in biology, and now we have come back for even more school. Yay! Because we just couldn't get enough. And we are in labs. So while we still go to classes, we spend most of our time in labs. We are in two different labs, so we're researching two different things, but we go to similar classes, and that's what led us to make this podcast. So I research DNA damage. So DNA damage occurs when things inside the cell or outside the cell come and damage the DNA through maybe double-strand breaks, single-strand breaks, and can cause things like cancer. So, Carly, what do you research?
It's pretty cool stuff, Kate. So in my lab, we are kind of centered around a few different parasites. So for me, I work on Plasmodium falciparum, which is one of the parasites or protozoans that causes malaria. So we work with lots of blood. Pretty cool, kind of scary, but I'm specifically looking at a protein that is involved post-transcription. So that has to do all with the central dogma, which I'm sure you all know about, from DNA to RNA and RNA to protein. So I'm kind of looking into those pathways as to what's going on so that we can hopefully find another treatment method for this good old parasite.
Very interesting. And I didn't understand any of it. What will this podcast be about? We are going to talk about different topics in cell biology and hopefully bring it down to an understandable level. We're going to talk about different things in the news, so looking at new research being done and connect it to its basic cell biology principles. So why is research being done and new research that is being done and how it connects to things you're learning in class. Today, we are going to be talking about LDL cholesterol and a new treatment that has been found for high cholesterol. So I'm sure you've all heard about good cholesterol and bad cholesterol. So when adults go in for their physical, they will go and get their cholesterol tested. And they're not really looking at their good cholesterol, they're looking at their bad cholesterol, which is their LDL. So they're not looking at the level of LDL in the cells, they're looking at the level of LDL in the blood. The cells have a method of taking that bad LDL, the bad cholesterol, and taking it into the cells and degrading it. But there are situations and diseases where that pathway is messed up in some way. One of these diseases is called heterozygous familial hypercholesterolemia. Oh, I think I said that wrong. Forgive me. We have linked the article and some information about this disease to this podcast so you can read about it. But basically, what this disease causes is high levels of cholesterol in your blood, which can lead to coronary heart disease. So it's not the high amounts of cholesterol in your blood that is bad, it's that it leads to coronary heart disease. Most people can control their cholesterol through exercise and eating good foods, but this research is focusing on the people who have a genetic disorder an inherited genetic disorder that prevents them from lowering their cholesterol. This research was done through genetic editing, gene editing. This research, this treatment is not a pill you take every day. It's not lowering LDL through taking in the LDL into your cell every day and lowering it and trying to make your cells take in more LDL every day, that kind of thing. It's actually changing your genome permanently. So now we're going to talk about how this genome editing was done.
So, I don't know about you, but I think that gene editing is so fascinating. I remember when CRISPR-Cas9 first kind of came into fruition, because women in STEM kind of helped bring that about with that Nobel Prize. CRISPR-Cas9 is relatively new, because we're so young, of course, we have to remember back in the day when that came out. So as Kate kind of said, CRISPR-Cas9 is a form of gene editing technology. This, of course, allows scientists, researchers to edit the genome of their target organism. I think it's kind of important to talk about how CRISPR came about. So CRISPR itself stands for clustered, regularly interspaced palindromic repeats. CRISPR itself is actually the name of these repetitive DNA sequences that were originally found in bacteria, and they contain some viral sequences within them. It's kind of said that this kind of helps the bacterium themselves with their immune response. Right? From our sources that we'll link below. Basically, they're saying that upon a viral infection, these bacteria are able to transcribe the CRISPR DNA sequences, so that's like those viral repeated sequences, into RNA. This then can compose parts of the bacterial immune system, which I really did not know that.
So wait, is it the CRISPR is part of the bacteria and then it can cut out the virus?
So, glad you asked, Kate. CRISPR itself, which is those repeats, kind of goes hand in hand with Cas9, which is a nuclease. It kind of acts to create these double-strand breaks in the DNA. So, you ready for this one? When a bacterium is infected with a virus, the Cas9 comes in, kind of cuts apart the viral DNA. This is called a protospacer.
Big word.
Big word. These protospacers are then stored within the bacterial genome. And then, obviously some cellular processes have to go on, and they're stored in the immune memory of the bacteria itself. So then, kind of like us, or our cells, whenever, this virus comes back in to try to attack, it can't. Because our immune system kind of already has that copy within it. So I thought that was kind of cool. So the Cas9 acts like a pair of scissors, and in order to specify where it is going to cut, it has these things called guide RNAs, and they're kind of the pinpointers, I need you to cut right there. So Cas9 will come in, chop it in half. Because CRISPR is able to do these kinds of things with Cas9, these create those double-strand breaks that I talked about before. It initiates a mechanism to kind of repair these breaks. So this includes the non-homologous end joining, or NHEJ, can't speak, NHEJ, or the homology-directed repair. So then these kind of act to help, I guess, bring back your DNA or the target DNA. And this is where, the gene knockouts, the knock-ins is what they're called, CRISPR knock-ins, where the gene editing can then bring in different bases or take them out, which is really cool And I'm excited to work on that in the lab at some point in my life. But I guess that kind of gets us into specifically with these LDL experiments that we were talking about before, this new, I guess, kind of CRISPR creation, if you will, called Verve 101.
We love the name.
It's incredible. If you guys have ever read any kind of scientific news or articles or anything, you'll see that some of the names of these techniques or proteins or enzymes are just incredible.
Yes.
Verve is awesome.
Which our professor likes to name proteins and things on our exams, crazy things. Right now we have sloppin. Globin.
Yeah, globin? Was that one too?
Globin.
Yeah.
Globin. But that's in step with the crazy names that come up with different proteins and genes. But it makes them easier to remember, which is nice.
Especially when we were just talking about this earlier, when the scientists name it after themselves.
Yeah.
Like, if I discovered and knew anything, I would absolutely name it after myself. 100%. Could you imagine you're reading an article and it's like, Kate!
The Kate protein.
The Kate protein. And then I would be like, oh yes, I know who created that.
So, Verve 101 is composed of messenger RNA encoding an adenine base editor and a guide RNA that targets the PCSK9 gene packaged within a lipid nanoparticle. So that was a lot of big words. A lot of nonsense.
Girl, for me too.
So basically what it means is that it has a guide RNA, so that is what's bringing it to the right place within the genome. That's what's targeting the PCSK9 gene. And then you have the adenine base editor. So that is what is bringing in the adenine and adding that and changing the single base. So what Verve 101 does is it changes a single base in the PCSK9 gene. See, we need a better name
Just
for
a
it.
bunch of letters thrown together, it feels.
It needs a better name.
Yeah. Let's start a petition to rename it.
Rename this gene. And by changing that single base, which is insane, that it's just one single base.
And it can cause such a difference
Yes.
in, what happens down the line. It's crazy.
Crazy. It causes this gene to not be expressed. And this gene its purpose is to basically eat up LDL receptors. So going back into how LDL is regulated in the blood,
I'm connecting things that we learned from class. That is so crazy. It impacts the receptors.
yes. So what they found is actually that some people naturally have this mutation. And all they have found within these people, it was an article from the New England Journal of Medicine in 2003 or 2006, so a while back.
Older, yeah.
That people with this natural mutation just have lower serum cholesterol. They just have lower cholesterol. And so that's why.
That is so interesting.
Yeah, so that's why they targeted this gene. They were like, well, these people have higher cholesterol naturally,
Right.
so could we make it so that they had lower cholesterol like these people with the mutation? And so what this gene does is it actually just takes the receptor away. So how LDL is taken out of the bloodstream is there is an LDL receptor. And so LDL will bind with the receptor, and it is taken into the cell through endocytosis.
Right.
It's then taken to an endosome and then a lysosome. So the lysosome is filled to the brim with enzymes. and it will break down the LDL into its base parts, and those base parts can then go to do things in the cell. And it is no longer, since it's broken down, it's no longer doing bad things. If there are more receptors on the cell membrane, then there are more chances for LDL to bind and then get
into the cell.
degraded
Right.
in the cell.
Crazy stuff.
Who would have thunk? A thing to highlight is that in this study, no study is perfect. So they did have some negative outcomes. So this was a small clinical trial of about 10 people.
Well, that's a relatively small sample.
Yeah, and it was 8 males, 2 females. So as we know, females are underrepresented in most clinical trials.
Not cool.
Not cool, guys. And so we just don't know a lot about the physiology of drugs and how they affect women. So 2 people did have negative outcomes. One had a heart attack, and one died 5 weeks after injection.
Oh, those are not good.
So they did claim that these outcomes were due to these individuals being already very sick, because they did do this clinical trial with people who already had coronary heart disease.
That's what I was going to ask. Did they pick a specific subset of the population that was already sick or had, something in common? Because it would be further unfair for them to have some healthier people and then some that are very, very sick.
So it goes into the ethics of clinical trials of who should get the drug first,
Right.
The most sick or the least sick. So if you do the least sick, then you might get better outcomes, but then you're depriving the most sick of a potentially life-saving
Something
drug.
that could work, right?
So they did choose the most sick, but then they did end up having some bad outcomes.
Horrible outcomes.
But the people who they did treat had significant reductions in their cholesterol.
That's very interesting.
Mm-hmm.
I guess a major future direction for them would definitely be to have more subjects in their study. Because although they did have some pretty crazy results, positive results, still have to test it on a large group of people.
Yes, so they did say that they want for their next stage to have healthier people with the same disease to be involved in the clinical trial.
Yeah.
So in order to see if they have the same bad outcomes, essentially, to see, kind of tease out if the bad outcomes were due to the coronary heart disease or the infusion.
It would be cool if they got, different groups, larger sample sizes than 10, to do another study. So they're not just comparing the less sick people with the more sick people from the study in the past, but something with both groups, maybe a group in the middle that's on their way to it being worse, and then they could compare. That's really interesting.
Mm-hmm. And another thing to highlight is this is a medication, this is a treatment, it's not a medication, I would call it a medication. This is a treatment that you only take once. It's an injection, I don't know if you call it an injection or an infusion, I'm not 100% sure how they administer it, but it said they give an antihistamine and I can't remember, steroid, they give other, they administer other medications before they give it.
Okay.
But you only have it once. So, because it permanently changes your genome. So, this is very beneficial for patients because a lot of times, so many people have high cholesterol. Any doctor will tell you it's very hard to get a patient to take their medicine every single day.
Yeah, well, it becomes tedious I completely understand that. But then again, if it's helping you survive, have a better life, you would hope that people would take it, but it does become like a chore.
Yes, you would hope everyone would take their meds every single day.
Take your meds every single day.
Take your meds. But most people just don't. And these are the meds that treat high cholesterol right now are like statins that you need to take every single day.
Right.
So this would be a really beneficial treatment that you just take once and it permanently changes your genome and permanently changes how the cell processes work.
Gene editing.
Gene editing, man.
It's so crazy because it's obviously implicated in other diseases and conditions as well. And if more research continues on these different areas, gene editing will change everything. It already is starting to. And just imagine where it can be in a few years if research with it just continues.
Yes. And so that goes into, there are more clinical trials going into different diseases for gene editing, like sickle cell.
I was going to say, I read an article that said CRISPR specifically, because, it's kind of what we're talking about. CRISPR, I think in some of the initial studies, was mostly used on patients that have sickle cell disease.
Yes.
That's a big one, too.
Yes, so sickle cell is a major debilitating disorder where your blood cells actually form a sickle. Basically, the cells catch on your veins and your arteries, and they get caught in capillaries because the capillaries are very small, and they get caught. And that can cause intense pain, and it can even cause strokes. There is a big push to try to cure this disease. The thing is, while gene editing sounds amazing, you can put a little guide RNA and it can find the gene and then make a little cut.
Make it sound so simple.
I know, it sounds so simple. But then, what if, that guide RNA, because, it's just an mRNA that matches to a region in a gene.
Right.
What if it matches to another region in the genome with the same sequence?
Complementary sequence. Yeah.
And then makes a cut where it shouldn't. What if that makes a cut on a promoter? A promoter is basically the start of a gene. So when it's activated, the gene will be transcribed and then translated, and then a protein will be made.
Right.
So let's say the guide RNA brings it to a promoter, a change is made, and that promoter is turned on permanently. And that gene happened to be a proto-onco gene.
Then you get cancer.
Right on the money.
Which is horrible.
Yeah.
But, when you go into research, this is kind of the process of thinking, right? You have to think, okay, it could have all of these positive outcomes, but there's also so many implications, and that could cause something negative to happen.
Yeah.
There's two sides, and you kind of have to weigh out your options. A lot of the times you do it because it's for the greater good of the research, unfortunately. Well, fortunately and unfortunately, because some of the outcomes are not on the positive side of what you would expect. But, cancer definitely, if those regions are turned on, they're not stopping. And that's how cancer happens.
I will say CRISPR has come a very far way.
Oh, yeah.
And the chances of it cutting in the wrong place and all of these what ifs happening are very slim.
But not none.
But not none. So it feels very big. So, and everyone is always oh, I'm not doing any gene editing to myself. Because what if? it does feel very scary. But that's because like we're used to medications that like you take and the side effect is like a headache.
Dizziness.
Yeah, dizziness.
Right.
The side effect isn't a potential side effect isn't cancer.
Right.
But
or at least you don't read that far into
yeah,
it.
you don't read
You see
that far into that giant packet you get.
right, because sometimes the symptom list, if you will, is pages depending on what is going on. And yeah, I know a lot of people just don't read that.
Yeah.
I think as scientists, we definitely probably read that. I like to know what's going on in my body before I take something.
Yeah.
Or after I take something.
Both. So after you've done it.
Both.
Yeah. So it's that debate that's going on with potentially using more gene editing for human treatment. And I think it's always something good to have in the back of your mind. But I think the best thing to do is to move forward with CRISPR, in my opinion.
Yeah, I know, at least for my research, I plan on some point using it down the line, because you have the ability to do these knockdowns, or these knockouts, that's kind of the beauty of it. You can do kind of whatever you want, and it can just tell you all kinds of stuff about what's going on in that cell. You do a knockout, you can really see the function or the potential function of something that you're looking at, or it tells you more about the pathways that it may take. And of course, that implicates all kinds of other cellular junk, if you will.
Yeah.
Or even these like knock-ins, which kind of almost like enhances, if you will. Or it can also add back stuff that you have knocked out in the past. It's just crazy, but you can learn so much about specific proteins, enzymes, and their pathways that they take just by editing one nucleotide base.
Yeah.
And something so small has such a drastic effect on a cell or an organism.
Mm-hmm. And you can really go down a rabbit hole and like really go crazy with it and look at all the research, and there's so much being done.
Yeah. I mean, as there should be. Something like this, as we've said, it could help kind of bridge some gaps for a number of diseases, whether that be cancer, even like malaria would be something. All of these different diseases that have some ways to go as far as treatment methods go.
Yeah. Okay. So, what have we learned? Hopefully learned. We've learned about LDL signaling, that pathway, and how LDL is degraded in the cell. We've learned about gene editing and CRISPR. And we've learned about this really cool new treatment for familial hypercholesterolemia. Thank you.
That wasn't bad.
I know, I think I might have done it. That uses gene editing. So all of our sources are linked below, and we will highlight the article that we got all this information from that you can read. Thank you for tuning in. If you have any questions, let us know. Yeah. Hope you liked it.
Yeah. Bye.
Bye.